Hello, welcome to the Thermo Fisher Scientific Showcase presentation. We're thrilled to have you here today. This presentation is on biomarker discovery through tumor profiling. My name is Mary Napier. I am the product manager within the newly formed Genetic Testing Services Group at Thermo Fisher. Let's get right to it. So how are you identifying cancer driver events? Of course, there are multiple paths to understanding cancer and multiple paths towards biomarker discovery through multi-omics, profiling RNA, DNA methyl, counting copy number, and overcoming challenges with FFPE. All these things need to be integrated in order for your goal of finding cancer driver events easier. So in this next slide, this is just a timeline of all the various technologies and how they've advanced in cancer assays. So way back in the 70s, we had karyotyping, so we could look at the chromosomal level, looking at aberrations, and this included, of course, copy number gains and losses and ploidy changes. As we went to the 90s, we took it a little further with FISH, and this was a faster assay, less labor intensive. In the 2000s, 2005 to 2008 was sort of the sweet spot for SNP arrays, and that's where you saw the HATMAT project turn into genome-wide association studies, which of course, another program called the Cancer Genome Atlas was also kicking off at that time frame. As those programs progressed, we discovered the need for LOH and copy number gains, and that's where you get the array CGH. As we've come into the later on, we've got low-pass sequencing in 2016. This, of course, can actually add diagnostic yield to array CGH because it captures more content and more variant types. And of course, more recently, we see the adoption of dual index NGS through hybrid capture or even amplicon sequencing. And this is a fantastic technique. It provides you high sensitivity and high specificity with low error rates. So why are we still talking about arrays in 2021? Well, of course, we're talking about arrays in 2021 because dual index NGS, even though it's a fantastic technique and it's going to really open up new areas of biology for us, in order to do routine testing for copy number, arrays really still have a place and often complement NGS. And in fact, our speaker today, Min Fang, who is from the Fred Hutch at the University of Washington, she is going to talk about how you can complement arrays and RNA-seq. So there are kind of two types of cancer. There's the M-class cancers, which have a lot of hotspot somatic variation. A lot of SNP profiling happens in M-class cancers. And then there's these C-class cancers where copy number detection, either gain or loss, a whole arm, telomeric imbalance, those changes are important. So it's these C-class cancers where arrays still have a presence today and can add value to your research. The TGCA data showed that 80% of all cancer samples needed to be characterized by both SNVs and CNVs. So of course, we want to see the full picture. Today, Dr. Fang will be speaking about her use of Cytoscan. And here is the Cytoscan chip. This It's a great chip. It's actually on guidelines for heme cancers. They feel it's future-proof technology with balanced copy number as well as whole genome coverage. And of course, it can be used across a wide range of sample types, from blood, fresh frozen tissues, saliva, FFPE. Dr. Fang also uses another product, which is called OncoScan. And OncoScan CNV has been around for several years. Many investigators use it, and it's kind of our flagship product for high copy number changes. It's a dual array workflow, and it can give you ultra high copy number resolution. And again, it's an extremely robust workflow, again, on the GCS 3000, just like Cytoscan. And just like Cytoscan, it uses the chromosome analysis suite, and that is actually a complementary software. And it's fantastic for FFPE. So people often separate Cytoscan for blood-based cancers and OncoScan for FFPE. So you can see that here's a publication from 2012 where a FISH panel would have missed 6 melanomas in a particular study cohort that OncoScan detected with that use of FFPE and ultra-high resolution copy number. The microarray workflow is direct and it's simple. It takes 3 days from sample to data, less than 1 hour for your data analysis. You have whole genome coverage in addition to high resolution on copy number. That high resolution on copy number plus whole genome makes an array workflow as a standalone or as a complement to your NGS workflows. And of course, the results can come from the routine samples you use in your lab, from blood, saliva, to FFPE, to archival FFPE. One great thing about arrays is often you can use a lower percentage to tumor to normal, which just the end benefit being fewer sample reruns on that precious biopsy that you have from the patient. I'd like to thank you again for attending the Thermo Fisher presentation. Without further ado, I'd like to introduce Min Fang. She's an MD, PhD, and a professor at the Fred Hutch at the University of Washington. She has served as a genetics lab director for 20 years. She is board certified in both clinical molecular genetics as well as clinical cytogenetics. Her current clinical service is as the director of cytogenetics department at the Seattle Cancer Care Alliance. She focuses on molecular testing on adult as well as pediatric patients undergoing hematopoietic stem cell transplants. And she is talking about an improved risk stratification for BALL or B-cell acute lymphocytic leukemia patients using chromosome genomic arrays and RNA-seq. Thank you, Dr. Fang.
Okay, thank you, Mary, for that kind introduction. I'm really pleased to be able to talk to my colleagues here regarding our research experience on improved risk stratification for B-cell ALL using chromosome genomic array testing and also touch upon targeted RNA sequencing. So actually the CGC working group for lymphoblastic leukemia published a very nice evidence review paper early last year of genomic aberrations of BALL. In this paper, they had a very nice, elegantly written introduction for those of you who are not familiar with BALL. So it includes how in children, BALL occurs more often than adults, and the incidence rate of the disease in the United States, as well as clinical manifestations, the flow markers, and patient symptoms, and also the clinical covariates that are critical for clinical management, including age, WBC count, central nervous system involvement, persistence of minimal residual disease, MRD, at the end of induction therapy. Of course, most importantly, are the genomic abnormalities. So the paper was structured according to the genomic categories listed in this slide. The authors wrote nicely in detail, summarizing all the current literature evidence on different categories here. And most useful to our readers are probably the summary table of table 1. It shows the prevalence and prognosis of BLL genomic abnormalities in both children and adults. The highlighted rows are those entities included in the WHO 2016 version. As you can see, in the pediatric population, the most frequent genomic categories are the 1221 translocation and hyperdiploidy. Both received, each one was seen in about a quarter of the pediatric patients. Whereas in adults, the most common category would be the BCR-ABL positive, Ph positive patients. What I'm trying to focus on today, however, are these 3 categories where you have Ph negative or Ph like and ICROS1 deletion patients, which are common in both pediatric and adult patients, as well as CRLF2 rearrangement. So for the ICROS1 deletions, because it's frequent and early evidence showed some poor prognostic association of this category, but subsequent literature is very inconsistent about the prognostic strength of that genomic category. So a recent paper from Europe actually was able to refine the prognostic significance of ICROS1 deletion by analyzing the effect of co-occurring deletion of the B-cell development pathway genes, many of these listed here. So they were able to define a special category, which they call ICROS1+. It's a new MRD-dependent, very poor prognostic risk category. So what they did is they took almost 1,000 pediatric patients from a prospective cohort of the European groups being from Italy and Germany. Then they looked at a copy number aberration of all these genes here. And then they saw the prognostic significance of this category. And then they replicated their findings in an additional 417 patients from the same cohort. So the definition of the ICROS1+ is written here. So it's not only the ICROS1 deletion, but also the deletion when it co-occurred with deletions in CDKN2A, 2B, or PAX5, or the pseudo-autosomal region 1 on the X and Y chromosome. But these should not have any ERG1 deletion on chromosome 22. So this group of patients conferred the worst prognosis in the pediatric cohort. So when they were trying to define the pseudo-autosomal region deletion, the PAR1 deletion, as many of you know, there are several genomic loci, including CSF2RA, IL3RA, and the CRLF2 probe. So these regions, they're only considering those deletions of the CSF2RA and IL3RA region, leading to the retention of the CRLF2 probe with association leading to the P2RY8 and the CRLF2 fusion. So that would result in overexpression of CRLF2, and that would be regarded as PAR1 deletion. To be clear, because it's a European study, they actually use the MLPA method for the copy number assessment. But in our study, we're using the chromosome genomic array testing using Cytoscan platform. So in their study, the limit of detection is generally 30% of the tumor cells, whereas the CGAT, the array testing can give you the limit of detection about 20%, as we all know. So they saw, as you can see shown in this slide, that the ICROS 1 plus category, shown as the gray line, they have the worst outcome, whereas those non-ICROS 1 deleted patients, shown in the blue line, has the best prognosis. And the middle line is those with ICROS 1 deletion, but not necessarily the ICROS 1 plus signature. And so the 5-year event-free survival is significantly different among the 3 categories as well. So accordingly, the relapse would be the most among the ICROS plus group as well. So interestingly, the researchers also found that prognostic effect is MRD dependent. What they defined as MRD negative or standard risk was that if they saw patients were they will consider MRD negative when they saw a flow percentage of the abnormal cells in less than 0.1% of patients on treatment days 33 and 78. All the rest of the patients would be either intermediate risk or the high risk. So only when patients, if it's MRD negative, there's no specific prognostic significance for the ICROS 1 plus group. But when the MRD positive, regardless of intermediate risk or high risk, you will see the significant poor risk of the ICROS 1 plus group. So these patients comprise about 6% of this pediatric cohort, and they had higher white blood cell counts at diagnosis. They were often positive for the central nervous system disease. They were rarely a 12/21 translocation positive, which is associated with favorable prognosis. Their prednisone response and MRD kinetics were worse than for those who lacked any ICROS 1 deletion. And this category is also associated with a germline GATA3 SNP variant shown here, and previously defined as a high risk for developing B other or PH-like pediatric ALL. So we wanted to ask whether that signature and the poor prognosis also happen in adult B-ALL patients. So I'm going to show you results of what we saw using chromosome genomic array testing using the cytoscan platform and the targeted RNA sequencing result using the FusionPlex platform. So this slide I have shown you before in previous CGC meetings, that although 922 translocation as a balanced translocation cannot be detected by array testing, but when you have a extra PH chromosomes, oftentimes seen in BLL, you can actually infer from the array data clearly because you will have a copy number gain and with clear breakpoint within the genes of the ABL and BCR. And of course, this slide also shows you how you can see focal small deletions of like CDKN2A and B. So for post-transplant chimerism patients, the array profile is more complex. The upper panel shows you in a typical pattern of a chimerism with half donor and half host. You will see 5 tracks typically, with regions of 3 tracks showing they are matching donor and host. So nevertheless, you can still identify abnormalities for post-transplant patients. For example, in this category of chromosome 7, you see the loss of the heterozygous track of the short arm of chromosome 7, suggesting the copy neutral loss of heterozygosity of the 7p. And the small focal deletions of ICROS 1 can also be detected, along with the frequently co-occurring T-cell receptor gamma deletion. And those deletions of the ICROS 1 region is usually very clear and clean. In B-cell ALL, it's very frequent that you see only small copy number aberration without other major events. So you won't be able to identify clonal abnormalities of such patients with any other type of assays like array, like karyotype or FISH, but only array can detect those small focal deletions or gains. Another advantage of using array is that you can pre-sort your abnormal B cell blasts. The beauty of BALL workup is that these blast cells are fairly robust and they can show clean results after flow sorting to enrich the abnormal cells. For example, shown here, a unsorted BLL patient with small gain, low-level gain of the XP, barely above the general line. But when you are able to enrich that population, then you can see a very clear increase of the copy number. So you can see that to analyze post-transplant patients when the blast count is very low. In this example, this patient at diagnosis showed a flow positive abnormal cells in 82.6% of the cells of the bone marrow. So we didn't need to enrich, and the patient showed a 9p deletion encompassing the CDKN2A and B locus. And a year later, at patient relapse, a patient was already post-transplant, and the profile would have been very messy, and the chimerism study showed 0 to 1% of host cells only. So you would have a difficult time to see any abnormalities. Therefore, we sorted the abnormal B cells, and we were able to see a very clean profile without the interference of donor cells. And then you have the same large 9p deletion. But the difference here compared with the diagnostic sample is that now you see a homozygous deletion instead of a heterozygous deletion. This represents clonal evolution. So for the same patient, when we look at the chromosome 7 profile with the ICROS1 deletion and other small deletions and gains, they look exactly the same at diagnosis and relapse. So confirming this is really the relapse patient. So when we summarize our identification of the ICROS1 plus signature in a prospective clinical trial, we noticed a prevalence rate of ICROS plus signature in about 21% of the adult BLL cohort that we worked on. This slide just shows the first 14 patients, how we looked at that signature. So we looked at individual genes of the ICROS1 deletion, CDK2A and 2B deletion, PAX5 deletion, EBF1, ERG1, and the PAR1 region. But to point out, the PAR1 region using array actually is not reliable because the sparsity of the probe coverage in that region. So this is the caveat using array method to define this category. Nevertheless, we were confident in identifying 3 of such ICROS1 positive patients. And so I also want to draw your attention to, we were able to look at some of the low hypodiploidy patients, and all these patients showed much higher number of deletions in their genomic profile than gains, which is very consistent with the genomic profile of BLL patients. So for BLL, hypodiploid BLL, it's a high-risk BLL category with either low hypodiploid BLL or the near haploid type. Oftentimes for masked hypodiploid BLL, you can see multiple copy neutral loss of heterozygosity tracks by CGAT, giving you a clue. So we previously published in this area, and I didn't want to talk about too much here. Rather, I'd like to focus on the pH-like ALL category. So they can be PCR-able-like. So basically, in the WHO classification of 2016, it's called out as those with translocations involving tyrosine kinases or cytokine receptors. So it was beautifully summarized in the 2014 New England Journal of Medicine paper by Roberts et al. So for pH-like ALL, you will have about like 22% of the able class fusion and 6% to 9% of EPO and JAK2 rearrangements respectively. And predominantly the pH-like ALL, 60% of these would be CRLF2 rearranged. Another interesting thing is I want to draw your attention to the bottom of that slide. A lot of the times you will see frequent deletions of the B cell pathway gene, like IGROS1, PAX5, EBF1, etc. It accounts for almost 90% of those pH-like ALL. And so these small deletions are common and often seen with CRLF2 rearrangements and inactivating mutations of the JAK-STAT or RAS signaling pathways. And so these recurrent gene fusions, so frequently seen, they actually, these fusions also induce small deletions at breakpoints that are detectable by array testing. So we basically showed you with the slides that I showed you before already. So we felt like CGAT can really be used as a surrogate for identifying pH-like signatures. And the targeted RNA sequencing can definitively identify gene fusion. So we know pH-like ALL is already called out in the NCCN guideline as a risk stratification category of poor risk. And in both the pediatric cohort and the adult cohorts, there's an age-dependent effect of the risk. So a prognostic risk. So it is both more prevalent among adults and older adults and, for pH-like BLL. Also, the prognosis is worse in those patients. Older you are, the worse prognosis they are. So it is encouraged by the NCCN guideline, the recent version, that gene fusions and pathogenic variants, especially pH-negative patients, being worked up. So we recently finished a study that we showed using targeted RNA sequencing is very effective for BLL patients. So we looked at 28 patients with 32 specimens, 4 of those patients with paired fixed cell pellets and cryopreserved cells to compare the results. And we were able to show targeted RNA sequencing can really effectively identify abnormalities. And so we saw detected fusions suspected by CGAT. So this is a profile of one patient showing a small deletion that only 40 kb in about 40% of the cells that suggests a STIL1 and TAL1 fusion because the breakpoint happened right in the middle of those two genes. And in this case, fusion was later confirmed by RT-PCR. And in both cases, of course, the targeted RNA sequence detected the fusion suspected. In this case, was a relatively large deletion, 8.4 megabase deletion of the 5q, with breakpoints located at the PDGFR theta and early B cell factor 1 loci, and leading to the fusion of these two genes. And that was also confirmed by FISH and the targeted RNA sequencing. And so this another case of suspected patients with fusion, and the array actually was negative. As I mentioned earlier, this detection of the CRLF2 and P2RY8 fusion is actually deletion. The deletion in the middle is unreliable. The PAR1 deletion is very unreliable to be detected by CGAT. So this patient showed normal karyotype FISH in CGAT, but abnormal fusion by the targeted RNA sequencing. So just to summarize, CRLF2 actually is shown overall in about 10 to 15% of all BLL cases, but it's very prevalent, like almost 60% in the pH-negative, pH-like BLL. And the partner genes can be P2RY8, as I just shown you. It could also be IGH and other ones, which are not quite detectable by this platform. So for those of you interested, you can look at this beautiful, elegantly written review paper in Blood. So overall, in conclusion, I've shown you the molecular profiling with chromosome genomic array testing and targeted RNA sequencing can improve risk stratification for BLL patients. And ICROS1+ signature is a newly defined MRD-dependent poor prognostic profile in both pediatric patients and adult BLL. We are actually compiling the prognostic data, the outcome data, to be shown in the ASH later this year. And despite the CGAT limit of detection of about 10 to 20 patients, sorting abnormal B cells allows very sensitive detection of clonal copy number aberrations and copy neutral loss of heterozygosity. And targeted RNA sequencing can detect balanced translocations missed by the routine CGAT testing karyotype or FISH, and especially the P2RY8 and CRL fusion. So we think, we hypothesize that combining CGAT and TRS can effectively detect major poor prognostic BLL risk groups with hypodiploidy, ICROS+ signature, and pH-like signature. So I'd like to acknowledge the fabulous Seattle Cancer Care Alliance and University of Washington cytogenetics team. They work so hard and do a fantastic job. And thank you so very much for listening, and I'm happy to answer any questions.